and why we have so many different species. But what DNA has really done for us is allowed us to very objectively and to great detail, again, determine how many species there are. So whether you get two taxa together, or they occur together, are they different or not? So you have, before you just had morphology to work with.
Yeah.
Or you had some very primitive DNA or genetic tools like protein electrophoresis or immunology or various other techniques that we had started to develop. But once we got to the point where we could do DNA sequencing en masse, and you could do lots and lots of genes in a fairly quick period of time, you could really refine that and get a genetic imprint of each organism and figure out, where the species boundaries might lie, and in a much more objective way. But the real thing, the DNA, the thing that I've studied a lot about what the DNA studies have done, which have really revolutionized this field. I'm really excited also about how they are going to allow us to reconstruct the evolutionary tree of life.
Yeah.
And that is something, I remember reading a book by Robert Solander back in 1970, and basically saying we weren't able to do sequencing at that time yet, but we were able to actually look at amino acid sequences and proteins and various other things. But he said, that the evolutionary tree of life is actually unattainable.
What does that mean?
Well, we could only estimate it, that we're never actually going to have the data to actually resolve it. And in fact, what DNA sequencing has done is given us the mechanism to actually resolve the evolutionary history of life on our planet. And we're going to be able to actually define, and we're beginning to already define, the actual evolutionary tree, which is, again, the history of how organisms on this planet have diversified and who's related exactly to whom. And it opens up a wide variety of avenues of research for us in terms of looking at evolution of disease genes, or looking at evolutions of organisms in certain places, or whatever you might want to do. But it really allows us to do something that I thought maybe in my lifetime would never happen, or could never be done, period. And that's actually resolve. Go back to what Darwin's dream was, which was to resolve the evolutionary tree of life. So
really quickly, what are we capable of here in-house, and where would you like to see ROM biodiversity, the ROM itself go with respect to DNA technologies?
Well, the ROM has always actually been, it's interesting, the ROM has been perhaps fortuitously in some cases, depending on who is leading individual departments, but the ROM has actually been at the forefront of looking at DNA sequencing and different avenues of it. So we've been able to be doing this for a long time. And one of the things the ROM has done right from the beginning is save tissue samples from organisms in its collections. And so in fact we have over, I think I looked at the total, was about 200,000 tissue samples, frozen tissue samples in our collections, from which we can extract DNA.
Extinct species?
In some cases they're extinct species that are now extinct. But typically they're ones that are extant.
Yep.
But we have this huge broad tissue collection. We've also been at the forefront, places like people like Alan Baker in the ornithology lab, and many others have been doing DNA sequencing right from the beginning, when it was really laborious and you had to do it with radioactive plates, and you were doing it for one small part of one mitochondrial gene, to the point where now we can, because of the DNA amplification, where we can make copies of the DNA. We're able to do it en masse and do it much more efficiently. We started with a big lab with a lot of big centrifuges, radioactive room, and so on, to several labs in the museum, which now have DNA sequencers, automated DNA sequencers, which we can use to generate a lot of sequence in a hurry from a lot of samples. So the ROM has been at the forefront of this. It's always expensive. We always had to push the envelope and how we fund equipment and that sort of thing to be able to do it. But in terms of our collections, and the big thing about the ROM is the tissue collections are associated with specimens in the collection areas. So if you get a tissue sample and you get an odd sequence from it, or you get an expected sequence from it, you can compare it directly to the animal that's in the collection and identify it immediately. And so if you get something that's new, you can go back and see what that new thing looked like in the collections, and that information goes back to the field. Cool. So the connection between the collections, the big frozen tissue collection that we maintain, and the fact that we have researchers here behind the scenes we have always been at the forefront of this technology, has made the ROM a real player in this field in Canada.
It's amazing, and I think this is a very unknown truth about the ROM, and we need to get this story out. So, quick follow-up then. Partnerships is something we've talked about over the last 8 or 9 Google Hangouts, and you know me, this is sort of my role here at the ROM, is to bridge us with the community. How do we include others in this goal to document the tree of life?
Well, there's actually a couple of different projects that we've been working on. The one is there is a big Tree of Life project in various disciplines, so the Tree of Life for birds, Tree of Life, the Tree of Life for mammals, and so on. And individual researchers participate in those projects. They contribute sequences, they contribute tissue samples, and all that information. The thing is, once you have a sequence, it's the sequence. You can put it into a database and save it, and it's available for the entire community around the world to use.
The whole academic community.
And it's required. So these days, if you generate a sequence and you want to publish it in Nature or Science or the Journal of Mammalogy, wherever you want to publish it, you actually have to deposit the sequence first in a publicly available database like GenBank, so then everybody has access to it. So we of course participate in those projects, and we've contributed many sequences to them. And of course contributed many tissue samples that other people sequence and then deposit that information for those genes in those databases. But we participated in these big initiatives like the Tree of Life initiative. Yeah. We've also participated in a project called the Barcode One, which is a different kind of a project, which is one where you're actually trying to generate one small sequence from one gene for as many species on the planet as you can to give us a very quick barcode or a sequence that would identify that particular species.
Two species.
And it's a kind of a quick and dirty method. But the problem that we have in science, in biology, and in my field of taxonomy and systematics, is that most of life is undescribed. We think of, we shouldn't go into biology because we know it all already. Well, in fact, we know far less. We're going to guess there's maybe 30 million species on our planet alive today. There's all kinds of, and many more millions which have gone extinct. But there are 30 million on this planet today, perhaps. We probably identified between 1 and 2 million of them, and the rest of them are unknown. And so how do you go, and we are faced with this massive habitat change that's occurring on the planet, and climatic changes that are occurring on the planet. The forests and so on that these species occur in are disappearing very quickly because of human intervention mostly. We're losing them, but we don't know what we're losing. So we go to the tropics, and you go to some place, and I've worked a lot in Guyana, for example. The forests are very remote. They've never been sampled for the most part in a lot of cases. And if somebody goes in and does a big forestry project and cuts down, 1,000 hectares of forest there, all the organisms that occur specifically in that spot will disappear and will never have documented. So how do you go about doing that really quickly? Figure out how many species are there.
The barcode.
You do the barcode. You do this. You do a barcode of a quick individual gene. You figure out, okay, I've found 40 mice in this place. I've actually got 7 classes of barcodes. I probably have 7 different species. And then I can go back. I've got the specimens. I can identify them. And then the next time somebody comes back in this area, they can take a small sample from some hairs from a tail, or they can take a little bit of blood, or they can some other tissue sample. They can barcode it. And they say, oh, that's the one that those guys caught back in 2012. It's identified as this particular species. I know what I've got.
Yeah.
Or I've got 3 things that are new. I better take specimens of those so we can identify those because we know there's something new from this area. And so it gives a very quick way of trying to document diversity in a particular area without having to go through the laborious process of collecting the specimens, trying to sort them out in trays, figure out which ones are which, based on morphology, which is a much longer process. Yeah. This gives you a quick way of pinning them down and saying, okay, I've got 3 piles here. There's this group of mice, that group of mice, and that group of mice, which are different enough that they might be different species. Let's start organizing and working through them. And
so for everybody out there, Paul Hebert over at the University of Guelph is really a partner in that project, and he's working hard. And we also have done a lot of stuff with Brad White and Paul Wilson over at Trent with their DNA Profiling Center.
Yes.
So there's a lot of good momentum here in Southern Ontario. But as Mark says, the ROM's really at the focus of it. I'm just going to take a moment to remind everybody. We're at Google Chat right now here live at the ROM in the Earth Rangers studio within the Shad Gallery. You can ask questions via hashtag ROM Hang, and I'm just going to give Ryan the heads up that we are cruising through this. So if there's anything specific, give me a heads up. And again, you can watch live via our website or on YouTube, and just Google Google plus ROM Hang. So I'm going to tweak this a little bit before I take the conversation in another direction. I'm looking back behind us, Mark, and we got a big bird here.
Yep.
You know the moa.
That's one of many moas, yes.
One of many moas. Extinct.
Correct.
On the cover of National Geographic last month. Do you know what for?
Yep.
You know where I'm going?
Go ahead.
Okay. So we have one of the best collections of passenger pigeons here at the ROM, anywhere. We have the moa. Alan Baker, as you know, has done a lot of work trying to get the DNA out of this thing. So we have a lot of intellectual property and expertise on this idea of extinct species in a lot of collections. The cover of the National Geographic was talking about the extinction, about bringing the saber-toothed cat, the pigeon, the moa, the mammoth back to life. I'm not going to mention my opinion on this, but what's yours?
It'll be difficult. It depends on how closely related the organisms are to ones you already have, that ones that are already here, and whether. So if you work with a mastodon, you may be able to introduce some mastodon genes and splice them in with with existing elephants. If you're trying to recreate if you're trying to recreate a dinosaur, you've got birds, which are living dinosaurs. But you are going to have a much more difficult time reconstructing something that's much more resistant.
So let me take it a step further, just for our lay audience here. Let's say there's a guy in Boston. Let's just hypothesize for a second and say that he has used some technology and some chickens and this, that, and the other thing, and he has a living passenger pigeon in his laboratory. Let's just assume. What do you think ethically about us creating flocks and flocks of passenger pigeons for the world that we're living in today?
Well, I think it's a fairly remote possibility.
Okay.
If you could recreate an extinct species, if you could get one back, I don't know why not.
Okay.
I mean, what would be the problem? I don't know. But I think the actual practicality of actually doing that is it's fairly remote. I think you're more likely to be able to do something like, for example, there's a species in the mammal in the southeastern U.S., the red wolf, which has basically been hybridized out of existence. It hybridizes with coyotes, and most of the existing red wolves appear to be coyote hybrids. So the question is, can you actually go back and through breeding programs, try to breed back to a more original red wolf? I think that sort of thing is more possible.
Yeah.
Trying to recreate the passenger pigeon is going to be much more difficult.
Fair enough.
So I'm avoiding the question, but
no, you're doing very well. And this is a nice segue because again, he's my boss and he's a research scientist and he's probably the best storyteller that I've met here at the ROM, as you can tell. But he also is our leader. And as he said at the beginning of the cast here, he's in a lot of meetings, and the ROM has a relatively new CEO, Janet Carding, 2.5 years. We have a new strategic plan. Embedded in that strategic plan is this concept of maybe reorganizing the ROM a little bit. I don't want to get into too much detail, but I mean, from your perspective, why was this necessary, and how's it going? How's the momentum going?
Well, it's interesting because one of the things that we did when we took polls of people that are visitors to the museum, both online and to the museum itself, they knew the ROM is an unusual museum. It emphasizes both world cultures and natural history. So we have great collections of rats, and we have great collections of European armor. So we have everything from soup to nuts. And the ROM is a very encyclopedic museum in that sense, and unusual because most places you would divide a natural history museum from a cultural museum. It has great advantages to us because, of course, cultures don't exist de novo outside of their environment, and it allows us to tell environmental and cultural stories together, which is more and more coming to the fore as the environment changes quickly over time. So that's great for us. But the problem was, and we have all these great galleries and so on that talk about various topics, again, fairly encyclopedic. So people, when they came to the ROM, knew that it was a great museum. knew that we had great stuff on display, but didn't know what we were all about. So they didn't know what we're good at, what our areas of specialty were and what they weren't.
Yep.
And so they didn't really know what to expect. They knew about ancient Egypt, so they go to the Egyptian gallery. They knew about dinosaurs, so they go to the dinosaur gallery. Then from there on, they're kind of going, well, they've got some other stuff, but I'm not sure what it is. What we're trying to do is trying to organize ourselves around our areas of strength, some of which reflect the original museums from the University of Toronto that were used to make up the ROM, and with additions. So we're trying to focus on 8 centers that we have or 8 focuses that we have within the collection, within the collections and within research expertise that really emphasize the highlights of the museum, what we're good at, what our strengths are, and give people really kind of a table of contents when they come either visit us online or at the front door. They know what the ROM's about. Okay, they got these 8 areas of specialty. I should visit something in each of these 8 areas. So there might be ancient cultures in one case, it might be biodiversity in another case, it might be earth and space in another case, and it might be textiles in another. But people will know we have these areas of specialties, that we have people working in them, and that they can come and see something unique and special out the ROM, both in terms of collections and in terms of storylines, that they would not see somewhere else. And I also want to be able to emphasize the fact that the ROM is not just a storefront. It's not just a set of galleries that talk about, that emphasize particular parts of the cultural story or the natural history story. But in fact, we have people behind the scenes. We do have a great research engine. We do have great collections. 99.9% of our collections aren't on display. They are research collections. They're in the back of the house. They're actively used all the time. They're actively studied, but they are not. You won't see them. And we have all these people behind the scenes who create galleries, who create public programs, who work on education modules and so on. But primarily what they're doing is actually fundamental new research, generate new information, new ideas, which we will eventually present on the floor. But we want to know people that there's a real substance behind the scenes at the ROM. That the ROM is Canada's great research museum, and it's an international museum too. And the fact that we work all over the world, and our mandate has always been to bring the world to Ontario. And so you could see things, and you could see storylines from anywhere around the world that deal with a wide variety of topics. But we do have 8 areas of principal focus, and we've got numbers of people who are working behind the scenes on those collections and who are generating new ideas.
So we've come full circle, because as I started at the top of the Google chats, I think our first one was April 2nd, we're launching ROM Biodiversity. So this is really our first center. Mark has been so helpful in helping us through it. I'm the managing director of this center, and we launched it on April 2nd. We got a banner on Bloor Street.
We do, yeah.
You see that? And so basically we wanted to soft launch the first center because we had some capacity around this. We had a managing director. We had a great deal of assistance from a larger curatorial team. We had the Shad Gallery and the Shad Gallery gift, which allowed us to roll through things. But we're ready, almost, to launch our second center. And that second center is ancient cultures. And if you don't mind, I'm going to drag somebody into our conversation. Rae, come on into our Google chat.
Hey guys.
Hi, have a seat right in the middle.
Great to be here among your specimens. I feel like I should watch for mousetraps.
So I won't get too involved.
They're not usually referred to as a specimen.
Well, I was thinking more of the moa, but you're very well preserved taxidermy.
Okay.
And animatronic.
That's right.
These two are hilarious.
This is better than Disney World.
So Rae Ostman, let me introduce you quickly, and I'll ask you a couple questions. We have Dr. Rae Ostman is the Managing Director of Ancient Cultures. You've been in Canada. We have 2 Americans here, don't we?
Yeah, exactly.
I'm also born in Minnesota.
Outnumbered, oh my goodness. So Rae, you've got a couple minutes. Give us the lowdown on what Ancient Cultures is up to, how you're going to plan this launch and what the activities might look like, and how things are going for you.
Fantastic. So Ancient Cultures includes our galleries, our research, and our collections related to civilizations of the past. And so those include the mummies that you mentioned, but we also have Greece and Rome. We have a great New World collection of Ontario artifacts, some things from Peru, from the Mayan cultures. And we will be launching, as you say, soon in June. We have a fantastic exhibition opening on Mesopotamia, which is one of the most important civilizations, really helped shape our world today. And so we'll have a lot of programming related to Mesopotamia. We'll also have a family weekend related to ancient Egypt in June.
So slow down one quick second. Mesopotamia. I don't think they're going to get that. What is Mesopotamia?
So Mesopotamia, what I hear most often is, I remember studying that in school. So Mesopotamia means the land between the rivers, between the Tigris and Euphrates. And it developed around the same time as ancient Egypt, so many thousands of years ago. It was one of the first cities, first examples of domesticated plants, of agriculture. Writing developed in Mesopotamia. There's lots of the code of law, lots of the foundations of our society, bureaucracy, meetings, 120 meetings a day. Thank you, Mesopotamia. We can really trace back to the origin of living in cities.
Okay.
And why do people organize themselves to live in cities?
And did they teach us anything about texting?
Texting, yeah. So
I was in a meeting recently, and I felt that was really quite cute. What's the connection there?
Sure. So the writing in Mesopotamia was cuneiform, and it was on small clay tablets with styluses. And so rather than using an alphabet like we have, they had a series of symbols that they would impress into the clay. And so this writing system, you might think perhaps was straight literature, perhaps they were recording historical events. Actually, most of this writing was business. And so keeping track of how many sheep you were selling to Mark and how many, things of barley I was importing and bringing into the city. And so really keeping track of business and keeping track of things. That's what cities have been about for a really long time.
And they still are.
They still are.
So what can we, I think I'm going to just ask you one more question, Rae, and then we'll go back to Mark. But what can we, as a contemporary culture, learn from ancient cultures? I mean, what's the relevance of studying ancient cultures to our contemporary society?
That's a great question. One of the things that ancient cultures gives us is it lets us look at questions and issues across many different places in the world, so different environments, different geographies, as you were mentioning, and across many different kinds of times. And we can look and see if we can see differences or similarities and see how that gives us insight. And so if we're looking at something like climate change and we're trying to understand how populations adapt, if we go back to the archaeological record, we just have a lot more case studies to look at and examples and sort of insight into what happens when climate changes.
I got a question?
No, I want Rae to talk about her Twitter.
Ah!
Sorry.
See, we're really casual around here. I'm gonna just tell our whole audience this. Ryan, you can just jump in. You can be a part of this. But
Camera shy.
The message we got was, you're on Twitter now, aren't you?
I am on Twitter.
What's the account?
At ROM Ancient.
At ROM Ancient.
Yeah. And so we've just started up on Twitter. We're trying to sort of set up a community. And so aside from having great archaeologists, technicians, conservators who work behind the scenes here at the ROM, we've got connections to archaeological colleagues all across Ontario, all across Canada, all across the world. And we want to involve the public that's interested in ancient cultures, interested in archaeology. And so Twitter gives us a chance to be part of one big community that communicate with each other. So love to see you on Twitter, love to see you contribute your thoughts.
What's your hashtag?
What's Twitter? Sorry.
It's not a mammal.
I was going to say that one of the interesting things about the Mesopotamia exhibition and how it came about. Why do we choose certain exhibitions? Well, they relate back to our centers of interest. So in this case, it's the Center for Ancient Cultures, and of course, we actually have Clemens Reichel, who's a curator and actually a joint curator. We share him with the University of Toronto, but he works on ancient Mesopotamia. That's what he does. We worked with him and the British Museum, so we're bringing a British Museum exhibition here. We're incorporating some of our own material and our own ideas into that exhibition, and Clemens will curate it for us and provides a base of expertise here on Mesopotamia. And in fact, Clemens is, I don't know if he's left yet, but he's going very soon to work in Iraq. Which is the center of ancient Mesopotamia, and he's actually going there to do field work. So he's been working in that region for 20 years, and that's really his base. And the ROM has a real history of actually working in this area. We've worked in Iran, we've worked in Iraq, we've worked in Syria, we've worked in Jordan, we've worked in Israel, in all of these areas in terms of looking at the archaeology of the Near East.
Cool. And a quick connection here for Twitter and Mesopotamia is Commander Hadfield, who is in space right now, as you know, on the International Space Station, has recently released a photo of Baghdad, contemporary Baghdad. And if you get a chance, check it out and think about what you're seeing from space and how big, immense Baghdad is. And think about Clemens interact right now. And when does Mesopotamia open?
June 22nd.
June 22nd. I think we've run out of time. Mark Engstrom, thanks so much for coming.
Oh, we didn't even get into what I do, though. We didn't get into my research.
He's got to be half an hour.
We didn't get into my singing mice. We didn't get into lemmings, right?
Your manly mustache.
My mustache.
Which was trimmed this morning, apparently. Dr. Ostman, thank you for coming out. We are trying to keep these to half an hour, but I think what we're getting from you, Mark, is you want to do another Google Hangout next week.
Sure.
And the week after.
Well, whatever.
I think we'll do the next Google Hangout in Mark's office.
No.
Oh, no.
Too much paper.
It's really clean. We're just talking about messy offices, and yours ranks low on the messy spectrum, I think. Anyway, I think we should probably round things down. So again, thanks for hanging out with us today. Our next hangout and our last hangout of ROM Biodiversity Month is next week with Amy Lathrop. Her and I are going to talk about herpetology. Our senior curator, Bob Murphy, is in China right now, so we're going to talk a little bit about what he's doing there. I'll be wearing a Toronto Maple Leaf jersey. I think Amy is wearing another team's jersey, so you may want to pay attention to this hangout because it may get into some politics around living in Toronto and supporting the Toronto Maple Leafs.
Well, if you'd have told me I would have worn the jersey, I would have worn the Charleston Chiefs jersey if you'd let me.
Until next time here on Google Hangouts, we're going to sign off. Thanks a lot.
Bye.
Bye.
Bye.